Okay, in the last video we talked about general male reproductive anatomy, and we got into the very beginning of spermatogenesis, or the production of sperm, which has three phases, the first of which is mitosis. And we talked a little bit about mitosis in the last video, but essentially you are making a genetically identical copy of a cell, and so you start with one mother cell and you end up with two genetically identical daughter cells. Well, the second process, which is unique to gametes, at least in humans, is called meiosis, and where mitosis takes one mother cell and produces two genetically identical daughter cells. Meiosis takes one mother cell and produces four daughter cells, which are all genetically unique. And to make four daughter cells, it requires 2 rounds of division. And mitosis of a diploid cell, which our cells tend to be, makes 2 diploid cells. Meiosis will take a diploid mother cell and make 4 haploid daughter cells. Haploid means only one set of chromosomes. Diploid means 2 sets of chromosomes. So we mentioned that you and most of your cells have 2 full sets of 23 chromosomes, one from mom and one from dad. 2 full sets means diploid, di like 2. After meiosis, you get haploid cells, which only have one set, 1 through 23. The processes of mitosis and meiosis are ultimately pretty similar. So what is it that happens during meiosis that makes the product so different from mitosis? Well, the big thing is, during the first phase of meiosis, we said that we have 23 pairs of chromosomes, right? 1 full set of 23 from mom, 1 full set of 23 from dad. Well, those pairs of chromosomes come together during meiosis. So you have, let's say, chromosome 9 from mom. Well, you also have a chromosome 9 from dad. Those are called homologous chromosomes, and they come together in a process called synapsis. And so each of those chromosomes has been duplicated, so it's 1 chromosome, but 2 copies of the DNA, two chromatids. And when it comes together, those 2 chromosomes now have 4 chromatids together, so we call it a tetrad. So this is what that looks like, right? Before we get ready for meiosis, we look like this. Again, we're simplifying with just 6 total chromosomes, 3 types. They all replicate until they have 2 chromatids, and then your homologous chromosomes, in this case, like your black and your gray are homologous, your red and your orange are homologous, your dark blue and your light blue are homologous. They come together to form these tetrads, and then not only do they come together, first of all, they will swap some DNA while they're together, very likely anyway. And then the homologous pairs will get pulled apart from one another during the end of the first meiotic division, during the end of meiosis 1. That does not happen during mitosis, right? During mitosis, it's the chromatids that get pulled apart. Here, the chromatids stay together, but the chromosomes that are homologous get pulled apart. So after meiosis 1, you end up with 2 daughter cells that only have 1 set of chromosomes each. They each only have 23 chromosomes. Now, all those chromosomes still have 2 sister chromatids, right? Which is not the case at the end of mitosis. But they each only have half the normal number of chromosomes. In other words, they are haploid instead of diploid after meiosis 1. Now, we still have to separate those sister chromatids so that each cell only has chromosomes with a single chromatid. And that's what meiosis 2 is for. Meiosis 2 actually looks a lot like mitosis, right? You take these sister chromatids and you separate them. They go to opposite ends of the cell, cut the cell in half, and now each new cell has chromosomes which each have a single chromatid. But they're all still haploid because during meiosis 1, we cut the number of chromosomes in half. Now, when these homologous chromosomes separate, again, you have a chromosome 3 from mom and a chromosome 3 from your dad, and you're only going to put one of them in any particular gamete, right? Because your gamete only has 1 set of genes, not 2. So which one is going to go in this particular egg or this particular sperm? Your mom's chromosome 3 or your dad's chromosome 3? Well, it's entirely up to chance, right? And just because your mom's chromosome 3 went into that particular egg doesn't mean your mom's chromosome 4 will. Could be your dad's chromosome 4. And so you end up with some mix of your mom's and your dad's chromosomes in the gametes that you make, and all of your gametes are different. And that's why siblings have different DNA, right? So that's independent assortment of these chromosomes. Just because mom's chromosome 3 went left and went into this sperm, or sorry, this egg, or either way, mom's chromosome 4 may not necessarily go that direction, right? Maybe dad's chromosome 4. And also you have crossing over, right? We've swapped DNA during synapsis, and that also causes some genetic variation. Now at the end of meiosis, we have 4 genetically unique haploid cells, but they're not sperm yet. Right? They don't have tails. They need to physically mature. And this process of essentially popping a tail and a cap on them is called spermiogenesis. I know that's confusing because this whole process is called spermatogenesis. This process is called spermiogenesis. So let's go through all of this again. You have to start with mitosis, then you move to meiosis, and then you have spermiogenesis. Mitosis is important because your stem cell is called a spermatogonium, and plural is spermatogonia, and I know that sounds like, a land far off in a distant ocean named Spermatogonia. But that spermatogonium, if it divides through meiosis to make 4 cells, that's great. You've made 4 cells, but that spermatogonium is gone, so you can't make any more after those 4 cells. So really, that spermatogonium has to copy itself through mitosis, and one of those 2 will undergo meiosis and make sperm. The other one will hang out, remain a spermatogonium, will copy itself again through mitosis, so that the other one can go on through meiosis. That's why mitosis is important first, so that you can continuously do this process over and over and over again. So you have a spermatogonium that goes through mitosis, makes 2 daughter cells. One of those daughter cells will remain a spermatogonium. The other one will become a primary spermatocyte. And a primary spermatocyte will undergo meiosis 1. During meiosis 1, your primary spermatocyte will undergo meiosis, which means those homologous chromosomes will separate to opposite ends and to opposite cells. And so the daughter cells, you will get 2 daughter cells from meiosis 1, will each be haploid, and they will be called secondary spermatocytes. So secondary spermatocytes will go through meiosis 2. Again, after meiosis 1, these cells are haploid. So secondary spermatocytes are haploid. These haploid secondary spermatocytes still have, though, chromosomes that each have 2 sister chromatids, and you need to separate those. And so we undergo meiosis 2. Secondary spermatocytes undergo meiosis 2, and you will eventually get 4 haploid daughter cells. And we call these spermatids. Again, these are not spermatozoa or sperm yet, right? They are spermatids. They are genetically the same as sperm, but physically they need to mature. That physical maturation is the 3rd process in spermatogenesis, and it's called spermiogenesis. And so if you look at this, we start with a spermatogonium, which will undergo mitosis, make 2 copies. One of these copies is a primary spermatocyte, which will undergo meiosis 1, and make 2 haploid secondary spermatocytes, which will undergo meiosis 2, to make spermatids. The spermatids will undergo spermiogenesis, get their tail, etc., and become spermatozoa. Congratulations, you are more or less ready for an ejaculation. So the main parts of a sperm are the head up here, the midpiece, and the tail. There's also technically a neck where the head and midpiece come together. The head is where the DNA is. That's kind of the business part of the sperm. On the front of the head is what's called an acrosome, and this is an important piece. This acrosome is full of enzymes that will help the sperm burrow into the egg, essentially, so that it can fertilize that egg. Without the acrosomal enzymes, you cannot fertilize an egg as a sperm. The midpiece is full of mitochondria that create ATP and allow for the tail, which is really a flagellum, to whip back and forth, or really it's kind of more of a corkscrew motion to allow the sperm to swim to the egg. So again, the swimming is important because you're delivering this DNA to the egg. This is a closer look at those seminiferous tubules that we talked about that you find within a testis. And these seminiferous tubules on the outside have spermatogonia, and the spermatogonia will undergo mitosis and make primary spermatocytes, which are a little bit further in. Those primary spermatocytes will undergo meiosis I and make secondary spermatocytes, which are a little further in. And by the time we're all the way in here, we have immature sperm that are finishing up spermiogenesis. Now you also have cells in between here, which are called interstitial cells. And while the seminiferous tubules are in charge of making sperm, the interstitial cells are what make testosterone, which is a pretty important function of testes as well. So this is an animation of a seminiferous tubule, which makes it a little bit easier to see what's going on here. So again, our spermatogonia are on the outside, and as we go further along in the process of spermatogenesis, these cells will be further toward the inside, and these are spermatids that are ready to undergo spermiogenesis, and these are completing spermiogenesis, at which time they will pop into the lumen and float away to the epididymis, where they will finish maturing. You also have, in between these kind of developing sperm, these larger cells that are called nurse cells, or sometimes called Sertoli cells. And these help sort of nutritionally care for the developing sperm as they go from spermatogonium to spermatid to immature sperm. All right, even sperm that are physically mature, they look like sperm, and they can leave the testes, cannot actually fertilize an egg because they haven't been activated yet, right? And there are two main parts of this process of activation, or capacitation, it's called. The first happens when the sperm are mixed with seminal gland secretions. That is when they start being able to move the tail. Right? Sperm are tiny. They are built for light travel, essentially. Right? They are just some DNA with a tail and some mitochondria, but they're not carrying a bunch of sugars so that they can make the ATP to keep the tail going. Those sugars are in the semen, which are produced by the seminal gland and also the prostate. And so the seminal gland secretion full of sugars and also some other chemicals that activate the sperm, that allows them to begin moving. Now the other thing that has to happen in capacitation can't happen until the sperm are exposed to the female reproductive tract. So the female reproductive tract is an environment which sort of leaches cholesterol out of the membrane of the acrosome. The acrosome, if you recall, is where those enzymes are that allow the sperm to burrow into the egg. And if you have a membrane that has too much cholesterol, it's too tough, you can't rupture that acrosome and dump those enzymes on the egg, and you cannot fertilize that egg. So a sperm has to spend some time in the female reproductive tract to weaken that membrane so that the acrosome will be able to release its enzymes so that the sperm can fertilize the egg. These are the two steps in capacitation, right? One happens when you're exposed to the semen as a sperm. The other happens when you are exposed to the female reproductive tract for a while. So let's review this pathway of sperm through the male reproductive tract, right? We are built in the seminiferous tubules, and then we move through the rete testis and the efferent ductules into the epididymis. And in the epididymis, this is where we become functionally mature. Essentially, you get your acrosome, right, which is necessary. You hang out in there. You are stored in the epididymis until it's time for ejaculation, and then whoop, we leave through this ductus deferens. And this ductus deferens goes up and over the bladder and back down. It's pretty long. And the last part of it is called an ampulla. An ampulla is just a thickened area that's surrounded by smooth muscle that will squeeze the sperm through, where they dump into these ejaculatory ducts. This is where they finally meet seminal gland secretions and prostate gland secretions. And that is when they are able to start moving their tail, right? That first part of capacitation. These two ejaculatory ducts dump into a single urethra. First the prostatic urethra, then the membranous urethra, and then the penile or spongy urethra. Now this is where, again, the semen or the seminal secretions joined together, and you can see the bulbourethral glands are where the pre-ejaculate is secreted before all this. And we'll talk about those fluids and much more in the next video.